Welcome, everyone. Hello, I am Courtney Cheetah, and I'm a program associate with Wild Farm Alliance. Thanks for joining us today. This is our ninth lesson of 12, and at Wild Farm Alliance, our mission is to promote a healthy, vibrant agriculture that protects and restores wild nature and to help bring wild nature back to farms. Just as a reminder, everybody is muted. If you're experiencing any technical issues, there are moderators in the chat to help, and feel free to use the chat to ask questions of the presenters as well. Our 2050 vision is to see 1 million nest boxes and perches on farms, hedgerows of habitat to the length of to the moon and back to help support all the different kinds of beneficials on the farm, and to restore riparian areas because they're the lifeblood of the landscape. And we really see avian pest control as being part of a toolbox for managing crops. Next, I want to share a new resource that we are building and invite you to join us. If you are an expert in sustainable pest management and would like to showcase your expertise for growers and potential clients, complete the Profile Builder survey. Your location will be featured on an interactive map with detailed pop-up information that enables growers to quickly find experts specializing in their crops and addressing their unique needs, and that's located nearby. So that's a really great resource that we're putting together that we'd love you to be a part of. You can find the link in the chat for that. We also have a new publication that is so awesome. It's hot off the presses and up on our website. It's the Managing Pests with Predators and Parasitoid Habitat publication. Check it out and learn all about SNAP, which is the shelter, nectar, alternative food sources, and pollen, and the proactive cultural practices that complement farmscaping measures, including how to create a blanket of attack against pests. There are 10 major crops that are grown in the U.S. in California that are covered, and all their natural enemies. So that's a really super cool and useful resource. Check that out. I'm super excited to dive into that deeper than I have so far. Today's webinar is promoting the soil's beneficial entomopathic microorganisms with Surendra Dara of Oregon State University and Scott Aki of Certus Biologicals. The next webinar, which will happen on February 11th, is providing habitat for predatory insects and wasp parasitoids with Ian Grettenberg from UC Davis and Michael Russell from Oregon State University. And please consider attending all of the rest of our lessons. We just got a few left. They're occurring about every 2 weeks between now and through March. If you missed one of the early ones, they've been really great. The recordings are all up on our website under the online courses under news and events. And we have continuing education credits available from DPR and ASA. And I'm going to pass this off to Surendra next. If you have questions, put them in the chat. And otherwise, I will let Surendra take it from here.
Good morning, everybody. So I hope you all can hear and see me well, right? Okay, very good. And first of all, thanks to the Wild Farm Alliance for inviting me to speak about my favorite topic. So I'm going to talk about, basically microbial control of arthropod pests using entomopathogens. You see the biopesticides here for disease and pathogen control and nematodes and weeds and all the other categories of pests. But the main goal here is entomopathogens here. What you see is bacteria, fungi, nematodes, and viruses that are intended to control arthropod pests. All these are soil-borne microbes, and they have evolved to survive in the soil, on the plants, and with other microorganisms or macroorganisms in the environment. So the first thing is these 5 categories that we have: bacteria, fungi, microsporidia, which are actually now categorized as a fungi, are a part of a fungal group. And then we have these entomopathogenic nematodes and these baculoviruses that belong to 2 categories: nucleopolyhedrovirus and granulovirus. So these are Then you see the QR code, like I have QR codes on different slides, which will, if you have a phone, if you scan, then you can see the full article related to, the topic I'm talking about. So each of these microorganisms have a different way of infecting the host. So imagine this as an insect host, a caterpillar. So bacteria and viruses and also microsporia, they need to be ingested so that they get into the insect system and then they do different things. So they go through the infection process. Whereas fungi, they need to come in contact with the insect. They can be ingested too, but they're more active through contact. The nematodes, on the other hand, these are microscopic worms. They actually enter the host through natural openings, and with the help of bacteria, they cause mortality. So you see different microorganisms have different ways of getting into its host. So it is important to remember when we use one or more of these organisms so that we use them for the right target. So the next one is to show how each of those actually infect the host. So Bacillus thuringiensis is a popular example of a bacterial microbe, entomopathogens. What you see here is the cell, it's a spore inside, and then there's a diamond shape. One is the crystalline toxic protein. That is the one that actually causes the mortality. So when it is ingested by an insect, the alkaline conditions in the gut activate the protein, and the protein is attached to some receptors in the insect gut. And then, basically what I'm trying to say is trying to show the point. So these are the spores and crystalline proteins activated or delivered into the gut, and then the protein is attached to the midgut wall and makes a hole. As a result of that, then the porosity causes the gut contents to get into the hemocoel, which is the insect blood stream, and the hemocoel, the blood gets into its gut. So the internal system is messed up, and then it causes septicemia and kills the host within 2 days to 3-4 days, depending on the host and depending on the strain. There are different subspecies of Bacillus. Some are specific to Diptera, some are specific to Coleoptera, and a lot of them are commonly used for controlling Lepidopterous pests. So this is typically the mode of infection for Bacillus thuringiensis. The next one is entomopathogenic fungi. So when you have the host or the fungal spore, this is the fungal spore. When it comes in contact with the insect, it produces this germ tube, and then the germ tube grows further, and then it produces a structure called a pressorium, and it releases some cuticle degrading enzymes and also uses mechanical pressure to get into the insect body. Once it gets inside, it multiplies and then basically invades the insect tissues and eventually kills the insect. So this is a wax moth larva, and this is the tan color is what it is supposed to look like. But when it is infected by by Beauveria bassiana, the fungus, all these spots are melanization. That means insect is trying to fight the infection by when the fungal spore gets in, it tries to melanize and block it. But it is when there are too many spores, then it can only fight so much, and then it eventually dies. So during the process, it turns kind of pinkish because of that is the nature of that fungus. It has those some active metabolites and other things that produce that kind of turn them into pink coloration. Then the fungus is emerging after it killed the insect, invaded the host tissues and killed the insect. It is emerging from the insect cuticle and producing those spores. And then, when the conditions are ideal, there is ideal amount of humidity and now that the temperature, then it profusely produces these thousands of spores that can be released again into the environment and continue the infection cycle. The next one is you see these entomopathogenic nematodes, mainly Heterorhabditis and Steinernema. These are two genera of these commercially available nematodes, and each of them have an association with those bacteria. It is actually the bacteria that kill the host insect. So these nematodes enter the host body through natural openings and then start multiplying, and then eventually with the help of the bacteria, they kill the host, and when all the resources are used up, then they get out and then start spreading. So then this is another way of like actively spreading the infection. And then the last part is the baculoviruses. As I mentioned, there are two major groups, NPV, nuclear polyhedrovirus, and granulovirus. When they are ingested by the host insect, they get into the midgut and then they're activated there and basically infect the cells and kind of liquefy the insect, and then the liquefied insect is kind of either because of the irrigation, sprinkler irrigation or rain or like wind, they kind of get dispersed into the environment and then cause spread the infection. Can you move the slide, please? So here is a photo where you see on the top different entomopathogenic fungi. The first three are Bagrada bug, if anybody remembers them. They were killed by these different species of fungi. And the last one on the top is Lygus bug. And these fungi belong to a group that these are, they can be mass produced and formulated into biopesticides to apply. But the bottom ones are a different group. Pandora, Erinia, and Entomophthora, these are a different group of fungi. They can't be mass produced because they're fastidious in their requirements to grow on artificial medias, but they cause natural epizootics and then kind of decimate the hosts under, when the host population is high and the high humidity and the environmental conditions are favorable, they can cause a significant mortality. So you have the bottom ones is naturally infecting fungi, and the top ones they're also natural, but they can be produced as a commercial formulations. And Scott will talk more about some of them. And the next one, please. And what you see here, on the left, you see nematode infection. You saw the bottom slide earlier. The bottom photo earlier, the nematodes emerging from dead larvae. And on the right, you see Bacillus thuringiensis killed larvae, and the bottom right are larvae that are killed by viruses. These virus-infected larvae exhibit a trait of moving to the top of the canopy and kind of sometimes get attached and then die there. That behavior helps the virus to spread. If they're infected on a lower level of foliage and then die there and fall off, then they have lesser of a chance to spread the infection. But when they go to the top and they're liquefied and then dripping, all the virus particles fall on the lower foliage, and then it increases the chance of infection. Next one, please. So here I just want to show the other kind of behavior or the relationship of these entomopathogenic fungi have with soil and plants. So far you saw that entomopathogenic fungi or other ones, they just kill the insect, but entomopathogenic fungi have been known to have a relationship with plants. So they endophytically colonize these plants, and then when the insects feed on a plant that has been colonized by these entomopathogenic fungi, the toxins in the fungi can indirectly affect the insects feeding on them. There have been several studies showing that when the insects are feeding, not from infection or not by being killed, but the indirect effect on their reproductive ability or their survival, and then other kinds of fitness issues. So there is an indirect benefit if you apply. If the fungus is in the plant, then it is not necessarily just to have to kill the insect, but it can also indirectly affect. So they can also antagonize plant pathogens. I have done some studies, and others have also done studies showing that these beneficial fungi can antagonize plant pathogens, and they can also promote the plant growth like mycorrhizae. They act as the extension to the root system and promote, like a plant growth by absorbing more, moisture and more nutrients and so on. So you see, when entomopathogenic fungi are used, they are not only killing the insects, but they are promoting plant growth, antagonizing plant pathogens, and provide additional benefits. The next one, please. So this is just to show where can you use entomopathogens in IPM. You see that on the green one, the microbial control, that is where you use. And it is always important to remember that any tool we use or any kind of approach, it works well when it is integrated with the rest of the pest management or agronomic practices. And the key here is the next key here is about successfully using microbial control is to understand the pathogens, understand the life cycle of the pest and its habitat, and use the right pathogen or combination of pathogens. If it is an insect that has chewing and chewing mouth parts, then viruses and bacteria are better suited. If it is an insect with the sucking mouth parts like aphids and hemipteran insects, then fungi might be better suited. And then knowing them well, and then using them at the right time and with the right combination of tools really helps increase the success of microbial control. Just if you have time, just give me a quick. It is a 2-3 question feedback, but if you have time, please do. And this is my contact information. If you have any questions about microbial control or anything related to this, please feel free to ask me. I'll be happy to answer later on.
Thank you so much, Sarna. That was really great. I have a much clearer understanding now of how these entomopathic genetic microorganisms work on pest insects and how they can work with IPM. Do you want to, let's do the poll questions next. We're going to launch those.
Sure.
So they should pop up on your screen. And if you just want to take a minute to answer those, this is for both the PCA credit and just to make sure you're following along with us. And then we'll give people a couple of minutes to respond to these, and then I'll have Surrender you go over them. It's so cool just to learn like how BT works, like having used it in my previous life as a farmer. It's cool to just get a better understanding on that microscopic level of like how it works. So cool. Surrender, do you want to go over these 2 poll questions?
Yes, yeah. So the first one, most people answered it correctly. Which of the following is not a type of biopesticide? Synthetic chemical pesticides are not biopesticides. And then the second one is a little tricky. I think, technically they can be dispersed by all like wind, water, infected host movement, and soil contact, all of them. So, I leave it up to the organizers which one they want to pick. Infected host movement is correct too, and then they can be dispersed by water and wind too. There are some fungi that are mainly dispersed by wind, some by water, like viruses I mentioned, and soil contact that is also there.
Wonderful. If there, I'm gonna just check here and see. Looks like there was one question about which fungicides might be the most compatible with the use of BTs. And it looks like Scott jumped in there. But Surrender, did you have any comments on that?
Which fungicide has most compatibility with BT?
Yeah.
I think most are probably compatible because BT is a bacterium and fungicides are not supposed to. But any fungicide can have an impact on any other microorganism too. Sometimes they're 100% compatible. Sometimes they're not. So it is best to check the label and see if there are any issues. For example, some of these fungicides are highly compatible with fungi as well. I did a study, and except for Captan and Thiram, rest of the fungicides I evaluated, they were compatible with the Beauveria bassiana. So a fungus and fungicide can be simultaneously used against an insect and a fungal pathogen.
Great. There's one more question about, could you briefly explain why viruses and bacteria would be better suited to target insects with chewing mouth parts?
Very good. So because they act in the gut, they need to be, they are better suited for insects with chewing mouth part because they chew on the leaf that has these bacteria or viruses. So, that gives them a better chance of ingesting these organisms and then, and get them to the place where they actually work. Whereas fungi don't need to be ingested because they're contact kind of materials. So that's why they're actually fungi are suited for both. Thanks for asking that question so that I could correct it myself. So fungi can be effective against insects with chewing mouth parts too, but they're better suited because bacteria and viruses don't get into aphids that easily. So fungi are a better choice there.
Great. I'm going to hold off on the further questions and we're going to jump over to Scott's presentation. Thank you so much, Surrender.
Thank you. I am back online and you can let me into my computer.
Perfect.
Thank you. And sorry again, everybody, for the mix-up.
No worries.
Okay, so thanks for the invite. I appreciate being part of this series. Today, I'm going to be separating out from what Surendra talked about. I'll be talking about the defense mechanisms that entomopathogenic fungi use to combat infections, what insects use to combat these infections. So first, we'll talk about the players. Surendra introduced some of these, and then the various defenses. So there's behavioral, physical, physiological, and then finally resistance. And then we'll go into some questions. So as far as the players go, there's the entomopathogenic fungi. So Bavaria, as Surendra said, that's a big one. Cordyceps, which is previously for a couple of them, an Isaria fungi, and before that it was a Pacillomyces fungi. So to be noted, they're all the same. It's just the gene jockeys have been reclassifying them. Metarhizium anisopliae is one that's getting to be commercialized. Metarhizium ecridium, it's commercialized. Lecanicilium, another Bavaria, another Isaria, another Metarhizium, and then a couple other fungi. So there's quite a few out there. People are actually surprised how many entomopathogenic fungi there are commercially available. The next group is the bacteria, primarily Bacillus. Other bacteria do infect insects. However, the Bacillus are the ones that are commercialized primarily because they form an endospore and it's easier to keep them for long periods of time without losing viability. So that's one of the reasons Bacillus has been very effective in the commercialized arena. Entomopathogenic nematodes, Surendra did a good job of explaining these, the Steinernevas and the Heterorhabditis. It's important to note that these actually do not kill the insect. They have a gut bacteria that kills the insect. So when we're talking about how they defend against infections. It can be for these, it can be either against the nematode itself or against the bacteria. And then the Microsporidia, which is now fungi and Nosema. This one is specific for grasshopper control. It's the only one I'm aware of that's commercialized, at least as of the last group I was looking at. And then the entomopathogenic fungi or viruses. There's two categories out there. There's granuloviruses, which is just a single encapsulated virus particle. And then there's nucleopolyhedroviruses, which they can be single or multiple viruses within a matrix. So we'll go into the defenses. So behaviorally, avoidance is one of the most effective defenses. So not coming into contact with the fungi, the bacteria, the virus, or nematodes. So there's been some studies done. This Antocorus nemorum is the omnivorous plant bug. It avoids outright plants that have the Bavaria bassiana on them. So it won't oviposit, and if it can avoid being present when the fungus is present on the insect that they're going to eat or on the plant, they'll not be around. They'll move to another food source. There's also these parasitoid wasps. Aphidus colomani is one good example where you can apply 1 kilogram per hectare of a Bavaria to plants that are infected with this or infested with this aphid, the cotton aphid, and allow it to, with multiple applications, allow it to just continue to be in these cages. And over multiple days, a couple weeks, you can see that the amount of infected or the amount of mummified infected aphids has not changed, whether it was not treated with the Bavaria bassiana or whether they were. So the parasitoid wasps, generally they move around a lot, so they aren't, and they touch the insects very minimally, so they don't have as high of a chance of getting infected. And then there's the famous example of antiseptic behavior, a defense. So honeybees, they did a study because these, as we commercialized these products, honeybees were of grave concern. So what they did was they did a field rate study outdoor under natural conditions. They put 5 times 10 to the third infective particles per hectare, applied it 3 times at 5-day intervals to these honeybees. They exposed the worker bees to it, and then they put them all back in the colony. They allowed the colony to function normally after they were treated with Bavaria bassiana for 45 days. And you can see over time, the mortality didn't change. The bees, there's a slight decline in the Bavaria treated, but it didn't change much. So in fact, there was only less than 2% of the treated bees that died compared to the non-treated. So they also did another study very similar. They came up with the same results. So why is this not happening? Why are they not dying when they're hit with an entomopathogenic fungus? So what's important is the bee body temperature is greater than 32 degrees C. Within the brood area of the hive, the temperatures are 32 to 37 degrees. Well, the upper limit where Bavaria bassiana can actually grow and thrive is 33 to 35 degrees. So the bees are not able to get infected because they're killing that, much like a fever will do to us. It'll kill off of the organism that's trying to infect us. In addition to that, bees also will remove infected individuals from the hive. So they'll actually preen them out and throw them out of the hive and onto the ground. So because of this, bees are actually being used to move Bavaria bassiana around different systems to infect other pests that we want to attack. So these are some other, if you want to look them up, there's some other behavioral defenses. Some of these are avoidance, some are antiseptic, but if you go through these, it's pretty important that there are quite a few systems where we do not, these biopesticides don't impact the insects. the beneficials. So physical barriers. The cuticle is by and far the best physical defense. The fungi actually enter through the cuticle, and they use a combination of pressure and enzymes to penetrate the cuticle. It's hard to. I mean, you guys have stepped on insects. You know how hard those cuticles can be. They crunch. So on top of that, this is my little joke here, but some ants will produce antimicrobials, and that will kill fungi and kill pathogens on their surface. And then there's actually some insects are known to have a biofilm, and that biofilm can actually impact entomopathogens from entering that cuticle. The other important thing is the digestive and tracheal system. So to note this, these actually have a cuticle because they're all part of the external integument of an insect. So they can be infected by entomopathogenic fungi. The issue is when you get into the gut of an insect, it's at a pH of 10, and a lot of fungi don't work at that pH, so it can deactivate them. However, things like Bacillus thuringiensis, it has a protein coat associated with this crystal. That pH of 10 breaks it down, releases the protoxin that actually gets that penetrates the digestive system and causes the septicemia. So in addition to that, you've got these viruses will do the same thing in that alkaline gut, that they've got a protein coat as well. They will degrade, the protein coat will degrade. It'll enter into the system of the insect. Rather than causing septicemia, it just hijacks the genetic material, the genetic machinery within the plant, and makes more virus. When it's all depleted all it can, it will just then, like Surendra said, those insects will just simply melt. So the tracheal system is a little less understood. However, it's a major way where nematodes can get in, entomopathogenic nematodes. There are other microbes that can infect in the trachea system, but they're still being understood. But it's, again, to note, that's also got a cuticle, so it is an area that's difficult to get into. A lot of these entomopathogenic fungi won't go right through the cuticle. They'll go through some of the areas that are weaker, the joints and such. So you'll often see sporulation in those areas. Physiological defenses. So we'll go through this quickly here. Once a insect sees that it's got a organism on the outside, it will recognize it as a pathogen associated molecular pattern, create these PRR recognizing acceptors, or it won't create them, but they'll recognize it. And it goes through one of two systems. It goes through the toll system, or it goes through the IMD system. IMD will do gram negative, toll will do gram positive, as well as other pathogens. And what these do is they just recognize that they're being under attack. It initiates the immune defense response, which results in antimicrobial compounds, enzymes, and also these hemocytes. The hemocytes are actually produced. A lot of times they're available already. The plant can, or the insect can produce them on demand, but there's three general types that are produced in these cellular defenses. One is a granulocyte. This granulocyte will go after a single microbe, encapsulate it, chew it up, basically utilize what it can and discard the rest. So it makes it non-infective. Plasmacytes they'll attack larger organisms. So what they'll do is they'll ensnare it, and then they'll encapsulate and cause nodules so that it's cordoned off from the rest of the system, so it can't be infective anymore. Then enocytes, they're a larger category, but one of the types of enocytes secretes antimicrobial compounds that will actually act directly against the microbe, killing it. And then Surendra talked about this melanization, but one of the general responses, one of the general physiological responses, which it actually encompasses these cellular defenses of hemocytes and other immune defense responses, is this melanization. So what this is it's a cordoning off of an infective particle. It darkens, it's a darkening pigmentation that aggregates around the pathogen. It isolates the pest so that it can't move or it can't cause more infection. And not only is it a physical, but it's also starving it for gas and food so that the entomopathogen cannot thrive and cannot infect. Some of these, such as like the enocytes, they produce the toxins, so it can actually kill the pathogen outright as well as it's melanizing around it. So resistance is a bigger thing. The most common known resistance in insecticides is to Bt because they were able to genetically modify plants. And also the cryotoxins are a little bit more simple nature so that their resistance can form a little bit more easily to them. In general, though, entomopathogens would be considered a multi-site active product, biopesticides. So compared to some of these single site pesticides, it's much harder to have to see resistance form to these. As I said, Bt, because of the wide use of the transgenic crops, they have seen resistance. And once you get resistance, it's a genetic predisposition to not be able to fight off these toxins or these entomopathogens. So since it's genetic, it's often passed from one generation to the next and to the next. So currently, we talked about the granuloviruses. There's a granulovirus used in the Northwest that we're seeing some resistance to. It's to control codling moth, but multiple 5 years of applications against this codling moth on a 3 to 5 day reapplication interval, we're starting to see some resistance show up. So the good thing with this is it is transgenic, so it goes from generation to generation. But if you take away this pressure, there's actually a fitness cost. So it will start to revert back and lose that resistance. So there's opportunities to overcome this as well. Unfortunately, with the BTs, it's a constant pressure on all these pests. So it's going to be hard to revert back to a non-genetic form of the pests that are showing resistance to the Bt toxins. So I hope I'm in time with my talk. That's all I have.
Great. Thank you so much, Scott. This is really fascinating. It's so cool to just get an insight into like what's happening on this microscopic level. We're going to launch the set of poll questions for Scott. And end it, and I will share the results. Do you want to discuss them, Scott?
Sure. So one, it could be any of those. The grooming behavior could be the bees with the antiseptic behavior where they're kicking out the larvae or the infected bees. That could be grooming. I don't think they do a lot of personal grooming. Secretion of antimicrobial compounds. There are ants will do that. Avoidance, as we saw some of those parasitoids, and the plant bug will do that. And an enhanced immune response. So pretty much everybody got it right.
Is there a most common?
The most common would be avoidance from what I've seen.
Okay, great.
So which of the following pathways can Bavaria bassiana use to infect a variety of insect pests? So Surrendra put this one through. It can go through ingestion because the integument is the same, but it's direct penetration through the cuticle. So
excellent. Then we can move on to question and answers. And if anybody has questions, feel free to put them in the chat and we'll get to them here. I want to go back to Surrender for a question about if you have any knowledge regarding fungus attacking fungi situations.
Yes,
No worries.
fungi attacking fungi means, I think, entomopathogenic fungi attacking plant pathogens. Is that what it means?
I think so.
Okay, yeah. Yes, like in one of the slides I mentioned, it attacking in the sense they kind of, you know, compete for the resources or release some of these microbial metabolites that can antagonize plant pathogens. So I did study with Macrophyllina in strawberry and Fusarium in cotton, and again botrytis in strawberry using entomopathogenic fungi. They, both Beauveria bassiana and Metarhizium antagonize these three pathogens. So they can, antagonize them. But the exact mechanism I did not investigate. Probably it is like multiple things that competitive displacement of these pathogens or microbial metabolites that antagonize them.
Cool.
I think it's important that some of your research also showed, Surendra, that in the absence of disease, some of these entomopathogens can be beneficial. Kind of, I don't want to call them PGPRs, but they
That's right.
do offer other benefits to plants.
That's right. Yeah, that's a very important point. If you remember that slide where I had the plant and these multiple roles, I wrote a review article about that. I encourage reading that article because these fungi need to survive in the environment when there is no host. The insects are not always going to be there, so they need to survive. So how they survive? They either produce, not all of them, some of them produce resting spores that can withstand harsh winters or like when there is no host, and others actually colonize plants, the roots or the above ground parts of the plant that gives them shelter, right? That ability to colonize or like have a relationship with plants gives them shelter and some nutrition temporarily. And in return, what they do is they support the plant growth uh, with more water supply and again, like protecting from pathogens and other microbial organisms. And they also transfer the nitrogen from dead insect into the plant. There are studies that show that when you have an insect in the soil, and then the entomopathogenic fungus kills that insect and also colonizes the plant, the nitrogen or nutrients from the insect are passed on to the plant. That is the benefit the plant gets by providing shelter to these fungi. So like Scott mentioned, there is we are not supposed to say certain things, but they do work like biostimulants. They are mycorrhizae and they're also pathogen antagonists and then entomopathogens like that kill arthropods.
So cool. I have a question for Scott. What about finding infected pests, mixing them with water, and spraying the infestation?
Hmm. I think one of the things that I've got actually to note that I forgot to was when you do have those insects, especially the fungi, if they're infected with a fungus, you won't see that sporulation under normal conditions. You actually have to put them in a bag, increase that relative humidity, then you see the sporulation. So I mean, it's possible, but we're talking in the range of 1 billion spores per milliliter that's going out in a spray. And so I mean, it's possible, but you would need to collect a lot of bugs, infected bugs. And I mean, I don't want to be flippant, but if you got that many bugs that you're infecting, then you might have other issues you need to think about.
Definitely. Are
So,
there
but
any?
viruses would also be, you could do that with viruses because they do. It only takes one occlusion body from a virus to infect another insect. So probably have a higher chance that way.
Sounds good. Are there any BTs that still work for DBM?
Yes, there's quite a few, and the commercial ones work for diamondback moth. In most areas, I should say, but there are areas where resistance is a problem. So there's the two types of Bavaria that are out there. One is, sorry, Bacillus thuringiensis, sorry. One is the Azawi strain, and the other is the Kerstaki strain. So if one of your strains isn't working, it might be worth trying another strain. I know there's a company out there that's actually mixing their Azawi and their Kerstaki commercially this year, and I think there has been some success against diamondback moth in those areas where resistance is there. But it'd be better to talk to your local guys about that.
And do you know how long it usually takes for the diamondback moth to develop resistance?
Man, Surrender might be better suited for that question. Too
Uh,
fast
it,
for a commercial guy.
yeah, it depends on the pressure, selection pressure, right? How they are exposed, and then the particular population mix. If there is no mix of wild or non-resistant susceptible populations in this one, then it takes time, takes longer. But if it is the same location and then concentrated selection pressure on the population, just a few generations is enough.
That's why the IPM strategy is so important.
Yes.
Getting something like a spinosad-based product or something other than a BT to reduce that single selection pressure is going to be important.
Excellent. And one more question, and if you have any other questions, please feel free to add them to the chat. But what about trichoderma?
It's
So
awesome.
was yeah
Awesome,
what
awesome fungus.
So trichoderma is more for pathogen disease management. Some strains of trichoderma can kill insects too. And then, all these organisms fight for their survival, right? Like, so there will always be some kind of interaction. If you put Beauveria and Metrrhizium, they will also compete. Which one infects first versus which one is late, right? And it depends on the host specificity to some extent and all those things. So trichoderma, if it can compete, but it is not a general tool for arthropod control. It is more for other purposes.
They have shown in a couple studies that trichoderma and some of the other fungi that affect plant pathogens in the roots do make a healthier plant. And so those insects, the insects that are searching for a host will often go by a color. Those healthier plants will be less likely to be hit compared to those that don't have like a trichoderma or one of the other pathogens that's attacking root pathogens.
Great. Jake has a question about what do you recommend for root knot nematodes?
Well, we're not supposed to talk about commercial products, so I'm going to back away from that one. Okay.
But
Very
but you
fair.
can
Yes.
but you can mention the fungus in the product.
There's one out there, Pacillomyces lilicinus, that's pretty good. There's a few strains that are out there.
So they
Great.
kind of suffocate the nematodes, like they're wrapped around the nematode and then suffocate and kill. And then there are also always this metabolite, like there are chemicals from plants and microbes and everywhere, right? Like, so they have their job too. Like, so it's a complex
These fungi will
mechanism.
directly infect all stages, all accessible stages of nematodes. The Pacillomyces will. So it'll get eggs, juveniles, and adults if they're accessible. So they're good. And trichoderma will. Trichoderma, they've got a lassoing type that will actually grow around a nematode and squeeze it to death. It's pretty cool. It's a little slow, but it's cool.
That's great. Love it. Well, thank you so much, Surrender and Scott, for sharing your knowledge. Thanks everyone for attending. We really love your feedback. And if you need those CE credits, we need your license numbers, and the DPR credit requires that you take a quick 10-question quiz. There are also additional resources up on the website that were recommended by Surrender, and Scott will make sure to get all the links that were in Surrender's PowerPoint up on there as well. And there's a great article on can insects develop resistance to insect pathogenic fungi that's up there as well. We hope to see you at our next webinar on February 11th. And thank you so much for attending.
Thank you, everyone.
Thank you. Bye.